Hello everyone and a warm welcome to all of you. My name is Yescarta and I work with the CAR TCR event series here at Hanson Wade and will be chairing the session today. Today's exciting event is the latest instalment of the CAR TCR webinar series, which is hosted by Lonza. The title of our collaborative roundtable is Bringing It All Together from Early Technology Evaluations to Commercial Approval. Our event today has been designed to take you on a deep dive into the lessons learned when evaluating and advancing cell therapies from translation to clinical trials and beyond. Our moderator, along with our expert panelists, will be discussing today's current manufacturing challenges and the future trends essential for developing robust manufacturing processes. It is my pleasure to now introduce you to Dr. Eitan Abraham, Head of the Personalized Medicine Business Unit at Lonza, who will be moderating today's discussion. Joining Eitan today for this roundtable event are three fantastic expert speakers. I'm delighted to have this opportunity to introduce Dr. Lawrence Cooper, CEO at Zyopharm, Dr. Isabel Riviere, the Director of Cell Therapy and Cell Engineering Facility at Memorial Sloan Kettering Cancer Center, and Dr. Stephen Feldman, Director of Manufacturing and Process Development at Stanford Center for Cancer Cell Therapy. During the webinar, there will be 3 audience participation questions. When the question appears, please respond within 10 seconds to the multiple choice. The poll results will appear soon after. Additionally, we will be taking questions from the audience in a live Q&A session. You can submit all your questions via the questions tab at the bottom of your webinar dashboard. Please submit your questions throughout the webinar. There is no need to wait until the end. We will endeavour to answer all questions in the allotted time. All unaddressed questions will be passed on to the speakers and a post-event report created with the answers. This will be distributed after the webinar, and if you would like access to this, please make sure you supply your email address when registering and opt in to receive emails from us. Without further ado, we will get started with this exciting session, and I'll hand over to Eitan to give us a brief introduction. If you could join me on the stage, Eitan.
Hi, thanks very much for the introduction. Very excited to be here. I'm sure we're going to have a very interesting and stimulating roundtable. We have some excellent panelists. I'm going to try to also navigate some things here. This is a new world. I have about 10 screens open, so we'll see how it goes. So as everyone knows here, or most people know, over the past several years, it's been very exciting in the autologous cell therapy space, specifically in the CAR T TCR arena. We've had multiple breakthroughs, we've had multiple commercial approvals of therapies, and also advance in solid tumors. However, it's clear that scalability, costs, raw materials, process robustness, to mention a few, are key concerns still in the industry, and we have to address these concerns in order to really make this space viable. So we're going to be discussing these and some other topics with our panelists. So I'm very excited to hear what they have to say to us. So without further ado, I'm going to invite the 3 panelists to the stage. Isabel, Steve, Lawrence, please join us.
Hello, good morning, good afternoon.
Good afternoon.
Morning.
Hello, everyone.
Excellent. So I think we have everyone. That's great. So welcome. You know, no one can tell where we are. I'm speaking from sunny Maryland at the moment. Maybe you guys can just say where you're speaking from, so it gives a little bit of feeling of actually being here. Steve?
Well, I'm in sunny California, Palo Alto.
Excellent.
And I'm in New York City, Manhattan.
Good. And I'm in Boston, and it's also sunny here.
Excellent. So we have a sunny day, and both coasts well represented. So thanks again for being here. So we're gonna jump right into it. So first of all, you guys are all very well acquainted with kind of how academia addresses developing these therapies and how they then transition into the clinic and hopefully and ultimately to industry and commercial approval. I'd like you guys to share some of your key lessons learned in your experience in setting partnerships up between academia and industry, and how kind of the path is to advance these therapies to the clinic and to commercial approval. Isabel, maybe you want to go first.
Okay, yeah, sure. So we have been working in this field for more than 15 years, and obviously the field has greatly evolved and progressed. Originally, we were able to perform research and development with grant money and foundation supports, and that's what we did for the early trials targeting CD19 for the disease such as acute lymphoblastic leukemia and chronic lymphocytic leukemia. So we were some of the first to launch these clinical trials using CAR T cells. But clearly it became really apparent that we needed to partner with industry and biotechs or pharma to be able to continue the launching and the later phase of these trials. So we have over time evolved in building partnerships with companies, and where an industry partner can support not only the clinical trial but also the process development, so that we can make progress and that we can test different systems and better systems in order to produce the cell therapy products.
Great, thank you. So really kind of a test bed for new technologies and obviously advancing various therapies to the clinic. Lawrence, do you want to give a shot at it?
Yeah, I think Isabel framed it really well. I think I've also been in the field about 15 years. Time flies. When we started this, cell therapy was squarely within academia. There really was no industry outlet that was really a catalyst. I think where that has now shifted is that the innovation engine now, I think, really exists both within industry and academia. I think there's a real balancing now. You see, terrific work coming out of great ecosystems, if you would. So I think that's one of the sort of the learnings that I take away over these years. And then I think that the other part is that both industry and academia have both embraced the concept that what we can create has to also be scalable. It has to be cost effective. Back when we started, it was so unknown whether this would work. We applied manufacturing principles, we applied learnings that were just really sort of, get it done and understand whether it will work. Now that voice is now changed to one in which, we know we can get it done, but we have to develop systems by which this is scalable, this is durable, this is cost effective. So I see those two kind of forces, innovation being now everywhere, and a real eye to essentially the ability to get this out into the hands of people who really need it, the patients.
Perfect, and obviously we'll expand on some of those points a little bit later on. Steve?
Yeah, I have to agree with my colleagues, but as being involved with one of the, I would say, major industry academic collaborations, that when I was at Surgery Branch NCI with Kite Pharma, right, similar to the UPenn Novartis experience, right? Those were, in my opinion, real collaborative efforts and partnerships. The companies needed the expertise, the innovation, obviously licensed the technology, right? So we worked together to co-develop these technologies, do a tech transfer. And then clearly at some point, the companies outpace the academics in terms of personnel and money and things like that. So, there was a point where, it was a feeling of that the company no longer needed the academic, in my opinion. And so, but I think like maintaining that partnership is really critical because, the academics can do a lot of innovation and have a lot of flexibility that maybe the companies don't have. So I think that partnership's important. And as Lawrence mentioned, now like a lot of the talent in the companies has come from academia. So it's a reverse now. The academics are looking to industry for the partnership because we need funding support. But what we can provide is some of the innovation and perhaps access to the clinic, right, which the companies don't have. So I think the partnerships are really important, will continue to be important. And I think the main thing I've learned is that it just needs to be a partnership. Don't just license the technology and disappear.
Yeah.
No, that's great. I mean, obviously, as you said, while things might be shifting this way or that, the academic industry partnerships continue to be key, and they're complementary things that each side brings to the table, which are very important for these complex therapies. So that's I think those were some really great points. Obviously, I think we're going to jump to our first audience question, which is surrounding kind of what people are thinking are the key considerations in bringing these therapies to the clinic and to commercial. So if we can pop that question up, that would be great. And everyone has about 10 seconds or 15 seconds to give this a read and make their choice or choices, two considerations. And then we can discuss what the panel's thoughts are on these. So first, while we're waiting for that, maybe for the panel members, in addition to these, which we'll talk about in a moment, is there anything else that you think is key that is not mentioned here?
Yeah, I think it's tough to choose one amongst the others. I think I'd be interested to see how the audience votes here, because I think all of them sort of matter. And they increasingly matter because as one, takes manufacturing capacity and scalability, thanks to the good work that Isabel and her team did, back, with CD19, that was, an amazing, proof of concept. But now the technology is, could have applicability everywhere, not just for hematologic malignancies, but solid tumors. So that's an important part of the consideration, is how do you get your proof of concept where you had one target to work for multiple targets. Cost of goods we addressed, really in the opening narrative, it's critical. Long gone are the days now where, these therapies could be produced, and delivered for, just eye-popping numbers. You can't achieve the scale unless you can get your cost of goods under controls. And the timelines, again, is one in which this is also really important, because patients' disease, has their own kinetics, right? A patient with CD19 malignancy is dying in front of your eyes. A patient with solid tumors may have some time with therapies, but there's no, often with metastatic epithelial cancers, there's no curative intent. It's one of chronicity and trying to keep the cancer at bay. So there allows for technologies to be developed that's commensurate with the patient's clinical narrative. So just opening thoughts there, really, as we sort of dive into this.
Right. No, great. So maybe we have some interesting results. So maybe we can pop up the results. So pretty clearly here, manufacturing capacity and scalability comes in at number one. Then timelines, and last in line, the cost of goods. And obviously we put these on because we believe all of them are needed. But it's interesting to see that still cost of goods, while being very important, takes a third place to the other two. So Steve, maybe you can comment on these results.
Yeah. I mean, I have lots of thoughts about this, but I mean, I always thought it's time versus money, and I think that time is more important right now for the patient, as Lawrence mentioned, but for the academic institution or the industry, the company as well, right? I mean, if you're developing a therapy, you need to get it to the clinic. And so that time is more important than the cost. I think capacity is a huge question, right? And there have been lots of conversations at various conferences and other webinar formats and panels around capacity and whether or not, you as a company, need to build your capacity or you can buy the capacity somewhere else. And I think for us at Stanford, we have capacity issues and we're looking to increase our capacity because it's all about, we're not just talking about one product, we're talking about multiple products, multiple clinical trials where we can support the manufacturing for that. And it's a huge issue. So the people are willing, it seems, to pay for the manufacturing facility because they feel like they can't access the capacity elsewhere. So I think capacity is a huge bottleneck right now.
Right. Great. Isabel?
Yeah. I totally agree with Steve that capacity is a big issue, and that that's something that we're going to have to resolve over time, because otherwise we will not be able to absorb the necessary number of patients that would require these therapies, and to make it affordable and also attractive for pharma to become involved and to take on the work. So, and personally, I think that working in clean rooms is not going to be the way to go in the long term. We're not going to be able to build thousands of square feet of GMP facilities with all the requirements and the infrastructure, as well as the operations that are required to maintain these facilities. So I think we really need to emphasize on the type of bioreactors that we should develop these cells in, and making them with small footprint, but which means also to improve the quality of the cells that we're developing in terms of efficacy, so that we can infuse smaller number of cells, obviously safe, and therefore with as little side effects as possible. But we want to make potent cells, and that's why also it's important, I think, in that innovation and development of improvement upon what is already out there in terms of anti-tumor efficacy of these cells is also very important. So I think we have to attack the problem from all different angles.
Right. No, absolutely.
Yeah.
Excellent.
And I would just add one other comment around capacity, and that is, we've also, as we think about scaling out, you're exactly right, the footprint, is one way, but it's also the people. These technologies, are so dependent right now on a skilled technical craft. It really is almost, artisanal in some respects, that we also, as an industry, need to think about, where those people are coming from, where those training programs are coming from, what type of operators are available to really grow and culture and nurture these cells. And it can be rate limiting.
We found, to your point, Lawrence, it is rate limiting, and there's a pool of talented people in the cell therapy space. They're either at industry or academia, but you're kind of cherry picking from each other in a sense, right?
Yeah.
So, we end up having to hire and train, right? And then those people will ultimately, well, they won't necessarily leave, but the probability exists. So, on the academic side anyway, we've invested in the training aspect of it.
Yeah, right. That's right. That's right. It can be a source. It could be a sore spot, to be honest, between industry and academia. In terms of where the people, get their training and whether they stay within the academic circles or whether they move on to industry.
Yeah.
And right. We're all in it together. So we don't want that sore spot, but at the moment it kind of stands out. Mm-hmm.
Right. No, clearly people are a key element, and I think you guys are at the forefront there as well, since you are the trainers in terms of the academic standpoint. So those are, absolutely great points. And obviously also, as we automate, we maybe can reduce the number of people that we need.
To Isabel's point, if we can come back to where we manufacture at some point, I think in a GMP ISO 7 space and ISO 8 space, or I think that's an important piece of the discussion. So maybe.
Absolutely, absolutely. Great. So I think we hopefully will touch on that a bit later. I want to switch gears a little bit and jump to the next question. So we all know that these therapies are different from traditional pharma in the sense that our starting material is different each time. It's a different patient each time we start the process. So what are some of the ways that you think we can account for or control that variability in terms of our manufacturing process and whether or not it's successful?
Well, I'll go first. I'll take a stab at it. I mean, basically, that's a difficult question, but it's a great question, right? I mean, because the product itself is a living therapy, biologic, and the patient obviously. So we can't fully account for all the variables, I think, of each individual patient. You can start like on the autologous side. You can maybe look at biomarkers from your apheresis or product and what expands in the patient and correlates with response to try and make products that you think will respond in the patient. But other than that, we've looked at the uniform starting cell product, doing a CD4/8 enrichment, say, versus using bulk PBMC. Maybe you can standardize the ratio, but I think the challenges are there. Or because it's each patient was different, or you move to an autologous allo setting. Sorry, there you can kind of standardize the product, but you still can't standardize the patient. So I would love to hear what others have to say.
Okay. Isabel, you want to try?
Sure. Yeah, I can definitely relate to what Steve is saying. I think the emphasis, in my view, in the autologous setting needs to definitely reside in the analysis of the cells which are potentially therapeutics in the mix that we're providing to the patients. And now with the downstream analysis of the cells that may be surviving in the patients and being able to correlate back to what was in the apheresis. Also, a number of groups have taken back their products that they have infused and stratified in them between the responses and the non-responders, and looking in the animal models as to what the various characteristics of these products might be. Doing some multi-omics, there is a number of biomarkers which are coming up, such as cytokine secretions or cell surface markers, some CCO7 or CD28, CD27, and others. And the question is how to standardize. And if we think that these are the cells that are more of an anti-tumor efficacy, how can we maintain and expand these cells? How can we select them first in the first place? And how can we expand them or genetically modify them in vitro in a way that is going to recapitulate what we think is the anti-tumor cells? It's an interesting exercise, and I think there are many steps, and they're not very good models to test the anti-tumor effects of these cells and outside of the patients. The animal models have some value. I think that at Sloan Kettering we're quite dedicated to these animal models, but the problem is it's a very expensive way and a very lengthy way of testing these potential new therapies. So it would be great if we can develop on-chip testing, and with potentially tumor models in 3D, and being able to test the activities of these cells in a much more efficient way. So that, and I think that's some areas where the community is working towards to develop these in vitro ex vivo models, but these are notably complicated.
Right.
Yeah, I think just to sort of add on to my colleagues here, the emerging technologies that appear interesting, are ones in which single cell analyses, at vast scale can be undertaken to look for individual actors, cell-cell interactions that are of interest, and then describe that in the aggregate using machine learning tools and very large complex data sets that can be understood for the human mind, and hypotheses can be developed. Those learnings around single cell analysis are really to get at what the key problem has been mentioned by my colleagues, is the heterogeneity of the product, the heterogeneity of the patient. And the great promise of living cells is that the complexity of the living cell is sufficient to handle the heterogeneity. And that's why I think autologous cell therapy will win the day, because when you're in an allogeneic setting, you're trying to program a cell for a defined operation. And at the moment, we just don't know, at least when we get into sort of the issues of solid tumors and the scalability for solid tumors, we don't know what those operators are. It's very difficult at the moment for immunologists to say what the form and function of a cell is. So we're better off, those biologists at the moment, using a scattergram approach, one in which autologous therapies, with all of their complexities of supply chain, but the one, to me, drumbeat that allows for optimism is that, when we make these therapies, there's a whole complexity of subgroups within the product that goes into that patient. Many of those are dead ends. Many of those cells will never hit the target or never be able to recycle their effector function, but some of them will. Some of them will. And then just to Isabel's and Steve's point, the learning cycle out of those cells that go in allow then for us to know that that was the right cell. It had a memory phenotype, it had a persistence, it had a trafficking ability, it had a killing function. And that is then the holy grail, because those learnings can then be taken back, just to everybody's point, to understand the production facility. How do you get that to work? But it's if you look, if you look at success, of where success has been in the field, and the two people on this call have had tremendous success, it's because we put in a rich mixture of cells. So so keeping a rich mixture of cells going, with and then understanding how they work. If we can keep that going, to me, that's when we're going to win here, because then we'll be able to outpace the cancer and really get to a point in which we can have effective therapies.
Right. No, great. That's fascinating, because obviously there's a debate about allo versus auto, and that's a very interesting perspective. Let's move on. So I want to go back for a couple minutes to our previous question, in a sense, and what the audience thought was most important, which is manufacturability, capacity, and scalability. But along those lines, maybe we can bring up our next audience question, because I think that goes to that point. And the question here is really assuming that we need to improve automation in order to scale and in order to reach the capacity, and, as was mentioned, in order to maybe reduce the need for a whole lot of skilled personnel. What's the right time to implement those solutions? So that's basically the question. And let's see what the responses are, but we can start discussing that. So maybe Steve, you can start. I see it's a pretty close race here, so that's interesting. But it seems like phase 1 edges out phase 2, 3, and preclinical comes in last, and that obviously makes some sense. But what's your read on that, Steve?
Yeah, it's an interesting question. I mean, I think the earlier the better, in a sense, right? If your plan is to use an automated closed manufacturing system, right? And so currently that's the trend. People want closed, functionally closed processes. Now it doesn't have to be automated, right? You can do that in other ways, but I think the sooner you get to a functionally closed system, the better. And I also think that, so I think that starts preclinical or process development prior to phase 1, right? Because I think in the phase 1, you are already starting to, the time frame for these therapies is shorter than other biologics, in my opinion. And you don't necessarily go to a phase 3 randomized trial anymore. It's like a phase 2, licensing trial. And so you need to lock in your process pretty early. And I think that, to the point that we had with the other question, it allows you to understand, the product and how it performs in the patient, because the process affects the product. I mean, we have lots of data around that now. And so to understand and engineer the attributes into your cell product takes time. And so I think the earlier the better is my opinion. So I think preclinical in phase 1 is.
So you're in the minority camp, but that's the camp I'm in.
That's the preclinical in phase 1.
Right,
so
absolutely, that's the camp I'm in too. So Lawrence, do you want to comment on that?
Yeah, I think the reason there's some sort of tension in the answers is that automation implies ossification. In other words, you've locked down a process to be able to put robots on it. So that doesn't necessarily have to be, and so I think, where the audience and perhaps, the panelists, might agree is if we had automated systems that allowed for the removal of the human operator, but still have allowed for some degree of, tinkering, then, I think we could really embrace technologies earlier and earlier in the life cycle. But of course, the issue is that at the moment, the robotics, the automation systems are one in which are really built to handle a process and do it at scale. You think about a fermenter, kind of thing, that, or some type of automated growth chamber or something. What cell therapy needs is an incubator, that you don't have to ever open the door. And one in which then, the human operator can be taken out of the equation, but yet still some degree of step change can be made, because we're all essentially living in a world in which the products we're making are first generation. And we know that additional work needs to be done. We know additional tinkering, if you would, has to happen.
Can I comment before Isabel? Sorry. I mean, I agree with you, Lawrence. Sorry. I mean, I think so functionally closed is important, and it doesn't have to be automated. I think if you go automated, right, you hit it right, the nail right on the head, that like scale down models to develop, like for the automated platforms, I don't know if that really exists at this point, and it makes it really challenging because the flexibility isn't quite there. So I just wanted to say that I agree with your perspective on that. And I think moving towards functionally closed systems is the key. Sorry, Isabel.
No problem. So yeah, I guess I just like to approach the issue from a different angle, and I think that when we, as we previously discussed, during a phase 1 clinical trial, we may still learn a lot about what is it that the active component in your cell mixture is, and that we may learn that a specific cell is a biomarker is present, and we suspect that it's an active ingredient in the process, and we may want to go back to the bench and to the process development and try to engineer these or reverse engineer these cells and test them in a new phase 1 or in a phase 2. So so I'm not certain that the early lock of the process in an automated way is necessary, because we still learn. I mean, in the phase 1 trial, we're still learning as to what the active ingredient might be, and what the next generation might be, and I'm not sure that pharma companies, because I think that's where, or biotechs may want to lock their process so early, because that may hinder your ability to make a better product prior to going to commercialization. So I think it depends somewhat of the what you predict might be the activity of your products in the early phase of the trials, and you may still be able to give yourself some abilities to evolve the product manufacturing prior to lock it in, and it doesn't mean that you won't have to do an equivalent or comparability trial, but I think that they may not be necessary to lock the process. I think it depends also on who you are working with, and who is supporting your trials, and when you are changing hands of the process from the academics to the industry, and what you think you're going to learn in the meantime. Go ahead.
So Isabel, I mean, I have a, so, you know, we manufacture using automated platforms at Stanford, and you know, we're not locked in, right? The challenge is that, you know, we're using the platform because it appears more efficient in terms of personnel, and you can train staff, and it's the footprints are fairly small, and less manipulations of the product. But you know, we're not locked in, and so we have to go back and develop. But so do you think, like, developing on an automated platform, because if we're doing phase ones, or you're doing phase one, and then it moves off to someone else who's going to do the phase two licensing, right? Then if you have to go and redevelop the whole process and do comparability and all that, seems also time consuming and a challenge. So what are your thoughts about just developing, I guess, on a
I'm just you know, if we're talking about timelines and how these products are developed, and I have always, almost without exception, experienced that we're being rushed on the front end to make a product with whatever is currently available, no time to do optimization of the process, and you know, whatever you may have learned last week from a better process cannot, you know, at some point you have to lock in. But I think during these phase one trials, you're still, I mean, I know, for example, right now, comparing bioreactors, simple bioreactors, which are available to everybody, and looking at the timing of the cultures, and you're using the same cytokines, the same activations, or you're comparing two different types of beads, two different type of media, and you still see serious differences in your activity of your product in whatever way you are evaluating your product during your process development. And these might, you know, indicate to you that, yes, right now in your phase one, you're using bioreactor A, but right now in your process development, you're testing bioreactor B, and your phenotype is much better. And so what do you do? I mean, you know, I think it depends on where you are, what is the competitive environment, and where the market is, and if you think that you're going to be able to develop this product or its alternative product in a timely fashion, so that it can be delivered to the patients, but it can also be viable for the market. So
right. It definitely sounds like, you know, there are obviously some tensions here. You know, move fast, do it cheap, you know, have an automated platform. It's also very amenable to making changes, right? At some point you can't have everything, but you know, I think it's, you know, this is obviously an interesting discussion as to when you jump. And I'd like to actually change things up a little bit here, and maybe because this is relevant to the discussion we just had, bring up the next audience question, because we've been discussing kind of, okay, when do you move to an automated platform? But then the other question is obviously, what are the main considerations when making the choice for that sort of platform? So is it cost? Is it flexibility? Is it comparability to the current process, or is it staffing, training, and FTE required? So let's talk about that for a moment, and then we're going to move over to a couple of other topics. So so the answers are coming in, and really based on our discussion, and maybe we can bring up the results. The flexibility is really number one, because while we want to automate and scale, you know, I think we're at a time in this industry where you really need to be able to have that flexibility, followed by you know, your comparability to your current process, and the cost of the equipment is really last here. So that's also quite interesting. So Lawrence, maybe you want to start and break that, you know, tease that apart.
I align with the respondents. You know, I think that's how I would have voted too. You know, the flexibility is critical, and I think you see that actually in some types of technologies that are now emerging in the automated space. You know, there is an eye to not only closing the system, but providing some operator parameters to be put in place. The ability to, the cost component being the other end of the extreme, being the least impactful, is also, I think, important to recognize, in that folks are willing to pay for this. You know, there's a real desperate need for these types of, platform technologies that are to be developed and put out there. And, again, perhaps, this Lonza, is a major mover in the field. This is something, for to take to heart here.
Great, excellent. Okay. Anyone else want to comment on this one?
No, I agree. So I would have put exactly the same order, and I agree with, it's important to have some flexibility of these automated system. I think being able to input parameters, and also I think from my experience working also in partnership with industry, having a modular approach in the manufacturing. I think nobody currently, because there are so few automated, do it, in one single instrument platform. Everybody is reluctant in investing really all their eggs in a single basket, and having no alternative technologies to individual part of the process. So I think the modularity also is important.
Yeah, I would agree with you, Isabel. Modularity is important. I think, so the interesting thing, right, as a developer in an academic setting in the phase 1 trials, right, where we manufactured using an automated platform, and then we want to make a change, a process change, because we start to understand our product, and we understand, that during the manufacturing, we're skewing the cells to a particular phenotype that may not be beneficial to the patient, and we want to change that. So then going back to make that change is challenging, because I think even Lawrence probably said, that the processes for the platforms have been established just kind of for a generic manufacturing. We want to make a T-cell product or something like that. But now when you get in the nuances, you want to make this product that, has a particular ratio, say, of CD4s and CD8s, or you want to have it not express a particular biomarker. Like, making those changes in an automated platform now is currently difficult, because what you do in the bench doesn't necessarily recapitulate itself in the automated platform, just because the kinetics are so different. So I think this is a real challenge. So the flexibility piece is huge. And so again, I think people that are in this space should consider that.
Right. No, that's great discussion. And obviously flexibility is key. I guess that, you can be flexible without necessarily being modular, but modularity is one way to be flexible. So it really depends on the specifics of what the platform is capable of doing. So I'd like to jump to a somewhat different topic, which is no less important, which is around raw materials. So we all know that we sometimes start with using non-GMP raw materials. Ideally, we use GMP. Clearly, when we get into later phase clinical trials, we need to use GMP and move to things like serum chemically defined media without serum and other GMP materials. So what are some of the bottlenecks that you've encountered there, and how do you approach that phase of using non-GMP versus moving to GMP materials? Who wants to start?
I can
Steve,
start.
please.
I always have no problem talking, apparently. I feel, so everything we do we try to source it as GMP materials. Although when you start to do some of this innovation and you want to introduce a small molecule inhibitor or something like that, or to skew a cell towards a particular phenotype or prevent exhaustion, or you want to use an antibody to enrich for a particular cell type, which may not be present at the end of the process. I think this is kind of the blurry area for me, where are those reagents ancillary products, and do they need to be GMP? And so, that's one question, but we've just sourced GMP material mostly from the beginning, because it just comes up as a question, because when you transfer this process, we move to out of phase 1, or the industry partners will all feel like everything needs to be GMP. So it's a good question. I don't have the answer to it, right? It's something we continually are up against, and does it need to be GMP? And I think there's flexibility in the phase 1 setting. So the other aspect is that you can just do what you do, develop your process kind of the fastest way to the clinic, in a sense, right? And then worry about it later. But it becomes a concern. We've had to regenerate reagents to make them GMP, taking that approach.
And any thoughts about chemically defined or serum-free versus media with serum?
Yeah, I think it's the same for us, right? We use human serum in our process, right? We use a chemically defined media with human serum, but we can get rid of the human serum, except that it changes the parameters, the kinetics of how the cells grow in that particular system. And now we want a 7-day process, or a shorter manufacturing process. Then serum is beneficial, but then we get the same question, like, do we need to remove the serum, as we move beyond? So currently, we have not removed serum from our process, but I think, and that's the trade-off, because we're about, a shorter process, because we think, maybe it's a more naive-like cell or a less exhausted cell. So we haven't quite sorted that out yet, but I think, others probably face the same question. But.
Any other thoughts on that one? Please, yeah.
I think that you can definitely, we also try as much as possible to use GMP, USP, qualified product, and then if we cannot, we interact with the vendors if these molecules are new or not available. And there are ways to qualify non-GMP products. It's pretty tedious. You have to really get from the manufacturers all the impurities, and sometimes they don't know, they've not measured them, or even if they know, they don't necessarily want to share the results. I've learned also to be, aware that some companies state that they sell GMP products, which are not GMP. So you always have to go and verify your certificate of analysis. And if it tests it passed, it doesn't mean that it passed your requirements. It passed the company requirements, which is not necessarily your requirements. So the quality assurance folks are sometimes a lot more doubtful than I am, and they're right. And it's really important to rely on them for the quality of the products. And I find that in the field of the antibodies, that's where we are really limited. Not being able to, some of the antibodies we would love to be able to use to select some cells, but unfortunately, the hybridoma story, at least for the old good antibodies that we like may not be, the history may not be available. They may have been exposed to all kind of sera and which were not controlled at the time, and I find that quite frustrating. So I hope that the industry is, and we have been approached by so many times by think tank leaders who want to what antibodies are you interested, but they can never jump the bar and re-engineer these antibodies and manufacture them under GMP. So I think there is a lag there, which it would be nice if at some point we have some real work groups who could really come together and define what would be the antibodies that would be really useful to the field, that could be developed and re-engineered and under GMP.
Right, great. So Lawrence, if it's okay, I'm going to jump to the next one. I'll give you first dibs on it. So one of the additional key questions right now is, do we go with viral versus non-viral gene modification with our products, and what are the considerations and preferences either way? So what are your thoughts on that?
Yeah, that's a good question for me in particular.
Right.
So yeah, look, I mean, I worked on a non-viral solution for gene therapy, for a long time, using a transposase system known as Sleeping Beauty, and the reason that I focus on this and is, I can see how this scales. I can see how, again, addressing some of the key issues of the field, how it is that we transition from, an industry which we're focused on one target, CD19, to where we can get other commercial opportunities now out there. In particular, where we need to make a product that has a collection of receptors, whether that's in what I'm particularly focused on is a collection of T-cell receptors that really target the neoantigen space. So as I think about that, and I think about going after solid tumors and having multiple products per patient, and those products being unique in many cases to each patient, then you need a gene therapy solution that can handle that. And so non-viral technologies such as the Sleeping Beauty system are one way to address it. And it allows for then the generation of plasmids that have, the ability to redirect specificity of T cells for multiple antigens, and the sort of production cycle for those plasmids is pretty fast, so that you can get your product identified, after the target, and you can make the product, all within essentially the life cycle that meets the patient's needs. So that's why I like it, and why we've really worked on it for so long, both in academia and now in industry, is because I think the non-viral world provides a solution to a problem. Not to diminish the viral world. I mean, again, these are everybody has sort of a niche in an area, but to me that's where the transposon systems really shine.
Can I ask a comment? So when you move in the non-viral space, and the transposon system is one system, and we've worked with you on this system. I think it's nice, but so making plasmid DNA GMP, right, is a little bit of a bottleneck. Maybe not quite the same bottleneck as making viral vector, but I guess my question that I have is, now if you move beyond transposon to say a gene editing platform where you want to non-virally insert DNA, and you need double-stranded DNA or linearized double-stranded DNA, or even single-stranded DNA, like how do you get that GMP, and does it need to be GMP? I guess is my question, because I mean, I think we're all moving. Viral works, it's efficient, but if we move to non-viral, like how do we do that?
Yeah, I think that's an excellent point, and it's kind of where we started our conversation an hour ago. Is things that started off in academia, back in the labs, when I was back at MD Anderson, that was one solution and there weren't the sort of the pressures of timeline and scalability and cost. But now that transposons now are worldwide, literally, there are companies all over, everywhere looking at this technology. So there now has emerged a group of vendors, who can manufacture DNA at scale really quickly. We're aligned with Aldebaran, and we find them incredibly helpful. The way in which they make DNA now is just a step change from the days back in academia, where they can handle essentially the complexities of personalized therapy on timelines, delivering multiple plasmids and so forth.
Great. So Isabel, maybe you'll excuse us this time, and I'll skip you on this one, but I'll give you first rights for the next one, which I think is a super interesting one, which is there's a lot of discussion about manufacturing at point of care / decentralized versus centralized, and you're in a very interesting position, as is Steve, in terms of doing these things at clinical centers. How do you think this is going to evolve in terms of the manufacturing locations, both for, I guess, late phase clinical as well as commercial? Are we going to move to point of care manufacturing in some cases, or is it going to really not happen?
Yeah, I mean, I think it's a very interesting question, and it's not an easy question to answer, but I think there are really some constraints. If you have a very large indication, I mean, especially in some of the cancer applications, I think it would be very difficult to have a point of care, because it would take up a lot of resources from the hospital to be able to treat all the patients that would come for this indication. And so even if you were to break it up into multi-center trials with a point of care, I think you would really put a lot of constraints in terms of operation and financing and manufacturing at the point of care for very large indications. I think for no matter what the platform might be. I think for the rare diseases, however, and where, in diseases which will not necessarily be taken up by a large pharma or a large biotech, this may definitely be a solution. And I would hope that this would be a solution, because I know that there is an unmet need for some of these rare diseases, and I think their individual hospital and point of care can be definitely of help, and should create consortia to be able to resolve these applications.
Great. Steve, what's your take on it?
Yeah, if point of care is in manufacturing within a hospital itself for patient treatments, I believe the main challenge, I mean, I think Isabel hit it, the capacity is obviously in space, but I think it's, if you have a really standardized process and the release criteria and the quality systems are in place, because that's a huge burden on a GMP manufacturing facility. I think in a hospital blood bank, say, where this might happen, I think that's the challenge that I see. So in addition to the throughput, it's part of the system, how do you release the product? And so, I mean, as it gets more universal and more standardized, it's possible, I think. But I think that's the challenge that I see.
Right, the release. Yeah. Lawrence, from an industry side of things.
Yeah. No, we've been thinking about this for a long time, and exactly right. It's a two-pronged problem to get to point of care, and that is, the manufacturing has to be, uniform, and the release has to be streamlined. So, that's really why, in fact, we have an IND for this and a trial at MD Anderson to test this out. In the CAR T space, is to do next day manufacturing. So again, with a non-viral solution, that might be quite interesting to then express a CD19 CAR using your transposon and then infuse it the next day. So solving for release on the same day of infusion and then using the transposon system to express a CAR and a molecule called membrane-bound IL-15 to try and essentially solve for problems of getting the cells to grow after infusion. So these types of trials that are underway, at Anderson. Also, we have a partner out in Asia to test this. Could be a solution to kind of then start to understand, to what my colleagues are saying, is this then useful then for industry to get to essentially localized, democratized manufacture? Basically what you're doing is you're taking a blood banking mentality. You're taking a machine and you're saying we're gonna gene transfer and infuse just as we collect blood and infuse. So it would be the same type of operations. But I think these types of solutions will be useful for industry and academia alike, because they get back to the question, at the beginning. What's some of the most important drivers for the field, and that's scale. That's, and it gets back then to automation, because it's a pretty simple process, point and click, really.
Right.
Great. I think that's a whole nother webinar, Lawrence. I think that's fascinating. And I would love how you characterize and release that product, next day. I think that's, we got to talk.
Yeah, I think the FDA would also be interested to hear how that works. But I think it's going to happen, but it's going to take a little time. Yeah.
And I was going to add actually that it's not only the manufacturing for the point of care, but it's the harmonization of the quality control and release criteria between the different.
That's right. That's right. And it's that learning cycle, right, that we're all in. We're in a rapid development testing environment, so that we can learn these types of biology and then feedback the knowledge, whether that's done in academia, whether that's done in industry. These are solutions that are all going to be important. Every one of these are going to be important, for as we go forward.
Right. Great. Excellent. So we're at time, and I maybe want to ask you as a last question to tell us maybe kind of what you find most exciting at the moment in the field, kind of where it's going in terms of going forward. We'll do a lightning round, and then we can close out. Lawrence, you want to start?
Yeah, I think the most exciting part is going after solid tumors. 1.5 million Americans every year have invasive cancer. We must start to use our talents to address this incredibly important opportunity.
Excellent. Yeah.
Yeah, you stole my line, but I think that maybe also the and the use of allogenic products and derived from iPSCs and other products, and which would be and other cell types coming online, not only with favorite T cells, but also NK and macrophages, and so widening the applications.
Yeah, I agree. We're all working towards solid tumor therapies, and so applying what we're learning in the hematologic space, with the CAR T therapies and others, I think, and applying it to the solid tumor TCR therapies, TIL therapies, or even CAR therapies. I mean, we're on the brink of really tackling solid tumors, so I feel it's just exciting.
Right. No, that's obviously super exciting, and it brings us back to the fact that at the end of the day, it's about the patients, and the patients don't really care how we make the stuff, as long as they can get the treatment. I think that's obviously an important message, and we're behind the scenes doing what we need to do to make that happen. So certainly very exciting times. So I want to apologize to the audience that we won't be getting live to audience questions, but we will definitely do our best to answer those questions and publish them, so that you have answers that whoever asked the questions. So with that, I want to give a huge thanks to our amazing panelists. I think it's been a really riveting discussion. For me, the time went by very quickly, hopefully also for the audience. So we've shared some of our experiences. Obviously, going back to Lonza, We'd really like to work with you to support your cell and gene therapy journey. Please go to our website to learn more about our cell and gene therapy portfolio and solutions, including the Cocoon manufacturing platform, our serum-free therapeutics media, our nuclear effector electroporation solution, the MODA software, and of course our CDMO offerings. So thanks everyone very much again for joining, the audience and the panelists, and hopefully we can have a follow-up discussion on some of the very intriguing topics that came up that we didn't have time to discuss this time around.
Yeah.
Great. Thank you.
Thanks very much, everyone.
Great, thank you all for taking the time today, and thank you again to our audience for participating in today's webinar. As Eitan mentioned, that is all the time we have, and we hope you have enjoyed this session. As mentioned earlier, all unaddressed questions will be passed on to the speakers, and we'll aim to share that in the post-event reports. If you do have any follow-up questions or comments that you haven't been able to submit in the chat during this webinar, please email these to myself at shaksita.desai@hansonwade.com. That's shaksita.desai@hansonwade.com. The recorded webinar will be shared with you all shortly. Thank you again for all your time, and I hope you enjoy the rest of your day.
Thank you.
All right.
Thank you.
Thank you.
Bye-bye.